It's a pleasure to be here, spending the day learning a lot about your funding agency and on other funding agencies here, and thinking about some of the strategic areas that are of great interest to my institute and at NIH, the National Human Genome Research Institute. This is a small group, and I knew it was going to be in a small room. I want to make this informal. I was asked to prepare about 30 minutes or so of overview, but please interrupt me, because what I did was to sort of take out a lot of the details of what I often talk about to make room for time, but I'm happy to drill down on anything people want to hear about, and we don't have to wait to the end. So really feel free to keep this as much a conversation as anything else. I thought what I would do would just be briefly set a context. I want to spend the bulk of my time describing sort of the present landscape of genomics, very much through the eyes of a strategic vision that we published as an institute on behalf of the genomics community a little over a year ago. And then at the end, I want to use that as a launching point to say a few things about the future. And I'm sure something you're quite interested, at least hearing some ideas of what we are thinking about in the United States, the future of genomics, and in particular in genomic medicine. Well, as you heard in the introduction, needless to say, I think clearly the starting point of all of this was the Human Genome Project, this large international unprecedented effort that I got involved in literally on day one as a postdoctoral fellow that aimed to, of course, map and sequence the human genome. It was an incredibly successful endeavor. On a mere 11 years or so following its inception, produced this draft sequence, the human genome, in this landmark publication in Nature, reporting that draft sequence and the initial analysis of it. The project, of course, still had work to do beyond that. It had to actually get a high-quality reference sequence of the human genome, and that indeed was accomplished and published, or it was accomplished and released in April of 2003, which then had essentially led to the final goal of the Human Genome Project, such a reference sequence. And as a result of the final goal being achieved, in fact, the project was declared successful and over. That was 9 years ago, or a little over 9 years ago. Now, one thing of interest is that at the National Institutes of Health in the United States, there's 27 institutes and centers. Our institute, the National Human Genome Research Institute, is one of those 27. We were created explicitly to lead the U.S.'s effort in the Human Genome Project. And so, needless to say, it sort of begs the question, of course, what do we sort of see our vision beyond the Human Genome Project? And of course, we care passionately about that vision. I give you various mission statements, but I would just simply state that our view of our role at NIH and actually in the world is to support research to help us improve human health through genomics research. And it very much is in that realm that we think carefully and critically and constantly about this idea of genomic medicine. And by genomic medicine, I mean healthcare tailored to the individual based on genomic information, not treating patients generically, but recognizing that each and every one of us, of course, has our own unique genetic makeup, and the differences amongst us will have very important implications for our health and well-being. And could we imagine a day where healthcare capitalizes on the uniqueness of each of us and tailors our healthcare based on some information about the individual's genome? So I've been involved in genomics since the beginning of the Genome Project. I've been at NHGRI for 18 years, but about a little over 2.5 years ago, I became the director of the Institute and really embrace this concept as being the core mission of what our Institute needs to do looking forward. And I sort of view this as a journey that the whole community and the international community is embarked on, but certainly at NHGRI, we think critically how we can contribute to this journey. So let me describe what I mean by that. I sort of think there's a path that we need to take that begins with the Human Genome Project. The Genome Project was immensely valuable. It wasn't the end of anything. It was really just the beginning of everything. And eventually we're going to realize this era of genomic medicine. But this is not going to be easy, and I don't want to lead you to believe it's going to be easy. It's going to involve a lot of different steps. In fact, we don't know what all those steps are. But we can go into this journey reasonably optimistic, having been very successful at the Human Genome Project, believing we'll be successful at realizing genomic medicine. And if we are successful, we'll have fulfilled the promise of why we sequence the human genome in the first place. So in thinking about this journey, the Institute has certainly been committed to it from the beginning. What transpired and thinking about this path from the base pairs of the Genome Project to the bedside, or if you prefer the metaphor, from helix to health. Literally, on the day the Genome Project ended, our institute published a strategic plan in 2003 that articulated the things that needed to be done in genomics to capitalize on the sequence of the human genome. And it was a very valuable plan, and I think it guided both our institute and I think more broadly the genomics community as the things that needed to be done immediately beyond the Human Genome Project. But what I will tell you more than anything is in those 9 years that have taken place since the end of the Genome Project and the publication of this strategic plan has been accomplishments in genomics that surprised even the most optimistic of us in terms of things that have happened. The pace at which technologies have been developed, the pace at which data has been generated, and therefore the number of new opportunities that have become available have surprised us and have really, I think, have taken us, and some of us actually, a little bit by surprise. You know, back in 2003, I think many of us thought of graphical images like this, where we could superimpose a DNA double helix and some imagery of clinical medicine and think someday, decades from now, this will be real. And we find ourselves now 9 years later recognizing that these aren't so crazy, these images, and they're actually on the near-term horizon and certainly within striking distance. So as a result of that, recognizing this incredible pace of accomplishment, we decided that it was time to update such a vision. And so last year we published this new strategic plan that much more directly embraced this concept of genomic medicine as the goal, and published it actually in the 10th anniversary issue of Nature that had 10 years precisely earlier published that draft sequence of the human genome. For those of you who have not seen the strategic plan and want to quickly go back to your offices and download it, and actually if you're interested in our strategic planning process, which actually was a multi-year process, and our community is very good at this, the strategic planning process, lots of workshops, lots of white papers, lots of input from advisors, and lots of debate. We actually catalog all this and captured it electronically. So this URL, you could get to the strategic plan, but you could also get to a lot of other information about this strategic planning process that you might find interesting. So what we heard, and I want to briefly introduce you to the framework under which we now think the strategic vision needs to be executed, was very much in response to what we heard very smart people in the community telling us, was that it was time to be more specific and more strategic in describing that journey from the Genome Project to an era of genomic medicine. And specifically what they said is we needed to be more detailed in describing the domains of research activity that would support in a very progressive way helping with this journey towards genomic medicine. At the end of the day, we found we could describe those domains of research activity into 5 major areas. And they end up being a very useful framework. In fact, our entire strategic plan is organized around these 5 domains. What are the domains? So the first domain, which was very familiar to us, is genomics research that helps us understand the structure of genomes, how genomes are put together. The second domain uses the knowledge of the structure of the genome to then start to explore how genomes work, so doing research to understand the biology of genomes. With that knowledge, one could imagine now being able to start to look at human disease and figure out in a genomic way what the underlying biology of disease is. And so the third domain is using genomics research to understand the biology of disease. And of course, with knowledge about disease in a more detailed level, it might provide insights for then being able to advance medical science. So using genomics to then advance the science of medicine. Although we were then counseled, don't stop there, because the temptation might be to stop there and declare victory, was the recognition that just because you have a medical advance doesn't mean when you actually go to implement that in the complicated milieu of healthcare systems, doesn't mean it's going to actually improve the effectiveness of healthcare, and that we would need, indeed, there's absolutely a seat in the front, feel free, to take responsibility to also recognize that we needed a research agenda in genomics that would demonstrate that we can improve the effectiveness of healthcare. So these 5 domains really represent areas of priority that we want to pursue in the coming years. The other nice thing about these 5 domains is that they provide you an ability to sort of inventory where you've been in the past and then where you might be going in the future. So let me talk about the past now, and I'll take you to the future later. The thing about the past, one way we found was useful was just say, at different time intervals, where have the accomplishments been in genomics? Just hypothetically, not precisely. And we could represent this by density plots. So here's an example, totally graphically rendered, not based on real data. But that by and large, during the Human Genome Project, all of the genome, the great majority of the genomic accomplishments represented by these blue dots were all about understanding the structure of genomes, how the human genome was put together, how other genomes were put together, what the order of the letters were, what the long-range organization was. And so you see the density of these accomplishments piling up within the first domain, and just a few things in the second and a couple things in the third domain. What about in the 9 years or so, or 8 years after the Genome Project? Well, I think it looks something like this, in that we continue to greatly explore the structure of genomes, but a tremendous amount has been learned about how genomes work and the biology of genomes, and indeed we've also learned a little bit about the biology of disease and maybe even got a few home runs out here in these more clinically oriented domains. So let me drill down a bit just to remind you what has been accomplished. There's another seat right here in the front, if you're, just feel free to sit down. Let me just go through some of these hypothetical accomplishments. Well, what do I mean to represent this interval of time? And actually, I think I could just briefly summarize these, as I will, sort of as 5 major steps that have moved us out from the Genome Project and have filled in this density plot over the last 9 years. So let me tell you what those 5 are. I'll just introduce each and just say a few things about them. It's one of the first things that we needed to do when the Genome Project ended. The first step, where we've seen a lot, is to understand the function of the human genome sequence. Genome Project was about producing a high-quality sequence. It was not about an interpretation. Interpretation of that sequence is going to take decades and decades. We have learned a lot in the last 9 years. We now know we have something like in the human genome 20,000 genes. We now know that there's a tremendous amount of our DNA that is getting transcribed into RNA, doing things that we previously didn't even know functioned in ways, involved in regulatory activities and so forth. We now know that it's not just the primary DNA sequence, but that epigenomics plays a huge role in conferring information from DNA, and methyl groups and histone modifications are all critically important. At our institute, we started a major effort, a consortium, international consortium called the ENCODE Project, the Encyclopedia DNA Elements Project, which is cataloging and developing very, very rich catalogs of functional elements, coding regions and non-coding regions, gene models, models about transcription, and so forth. And that project will continue because we recognized that there'll be decades of work ahead of us to fully interpret the human genome sequence. But by and large, we've really capitalized on these first 2 domains of activity by exploring more about structure of genomes to understand genome function, and increasingly used efforts like ENCODE to better understand the biology of genomes, but recognizing we have still a long way to go in doing this, but this was an important first step. The second thing we needed to pursue recognized the fact that we are not just interested in how a hypothetical human genome sequence functions, but we're interested in how each of our genomes uniquely functions, and eventually we're thinking about how our patients' genomes function. In other words, how are we different? And so another key priority for the genomics community in the last 9 years, and continues in the present time, is understanding human genomic variation. Keep in mind, across, each of you has about 6 billion base pairs that constitute your genome. You got 3 billion from mom, you got 3 billion from dad. And across those 6 billion letters, there's probably about 3 to 5 million places at a nucleotide level that you are different compared to the person sitting next to you. And those 3 to 5 million variants represent positions that are not unique just to you, but in fact, many of us share, in fact, all of us in this room share thousands of variants that are very common. And across the human population, most of your variants are shared, but some of them probably are very rare, maybe only in your family. But we can catalog that. That becomes a finite problem. And of course, we're interested in cataloging that because most of the variants in the human population have no phenotypic consequence, but a small subset do. I mean, some of those confer risk for disease. Some of those are protective for disease. We want to know that. So through efforts that you might have heard of, more international consortiums, you get the idea. Genomics, we love these international consortiums. The HapMap project you might have heard of. It actually followed something called the SNP Consortium, Single Nucleotide Polymorphism Consortium. And now most recently a project called 1,000 Genomes Project, which is a name that's not even accurate because we're studying 2,400 genomes. These are both for the HapMap project and for the 1,000 Genomes Project, taking discrete human populations from different geographic origins and collecting hundreds of specimens from each or dozens of specimens from each, and making these available, cell lines made, worldwide distribution of these cell lines, and getting lots of data about the variants that exist in each of those populations. Importantly, putting all that data on the internet so everybody can get to it, and having this be a worldwide resource of known, at least more common, genomic variants. And that's been incredibly successful and continues to the present time. As better technologies become available, of course, it gives us better and better ability to drill deeper into genomes to get more people, to get more and more information about rare and rare genomic variants. And why do we want, and of course, this still is within these first two domains of structure of genomes and increasingly try to understand how that influences biology. The third step along the way gets us more into this middle domain, because the third step that has been started is about now using knowledge about variants that exist out in the population, figuring out which of those might have detrimental phenotypic consequences, such as conferring risk for disease or overtly causing disease. Here, there's been really remarkable progress, although as much as anything, it just sets up what's going to happen over the next decade. And let me just drill a little deeper on this, the third of the fifth step, to remind you that all human diseases have a genetic component, either an overt genetic cause or at least some genetic influence. But genetic diseases can, by and large, be divided into two broad categories, depending upon their architectural feature. On the one hand, you have rare genetic diseases, diseases like sickle cell and Huntington's disease and cystic fibrosis, where it's a single gene that's defective that represents the dominant risk or the major risk for getting that disease. Sure, there might be some other genetic variants floating around that might influence the severity of disease, and maybe there's even an environmental influence. But by and large, these are simple, monogenic, involve one gene. They're also known as Mendelian disorders because that's after the famous geneticist Mendel. The thing about rare diseases is that they're not major healthcare burdens worldwide. They're horribly detrimental to individual patients and their families, but it's not what fills hospitals and clinics around the world. This is what fills hospitals and clinics around the world are common genetic diseases: obesity, hypertension, diabetes, cardiovascular disease, asthma, and so forth. The problem with common diseases is that they're genetically complex, because typically it is multiple genetic variants that together conspire with what is typically a greater contribution of the environment to then confer risk for getting that disease. Well, the real question, even when the Genome Project ended, was we already knew when the Genome Project ended that indeed we were going to accelerate the pace of identifying the genetic causes of rare diseases. And the reason we already knew that is that when the Genome Project began, there were about 36 genes that had been identified causing, when mutated, a rare genetic disease. And by the time the Genome Project ended, that number was well up into the hundreds. But what's taken place since the last 9 years is that that number is now 3,500 or so. We now know the genetic basis of 3,500 single gene rare disorders. And that's spectacular. And we'll come back to that when we go to talk about the future. We knew almost all along that the Genome Project would accelerate rare genetic disease research, but there were lots of questions about whether it was going to work for this complicated situation where you're going to have to tease out at a statistical level an individual variant that doesn't cause a disease, just influences it, and does so by conspiring with other variants and the environment. And what if I was going to take a huge body of literature and summarize it, what has come to the fore in the last, in particular, 5 years, has been these things called genome-wide association studies or association studies, whereby you take large sets of people with a genetic disease or a complex disease like hypertension and 1,000 people without the disease, and you do this complicated genetic analysis that allows you to find statistical associations between discrete regions of the human genome and getting increased risk for getting that disease. And where once upon a time it was a back of the envelope, maybe it'll work, maybe we can make association studies work, has been a resounding success. And with respect to the studies themselves, fundamentally are giving us great clues about where to look in the genome. Give you some numbers. The first successful genome-wide association study was published in 2006. Now there's over 1,200 studies that have been published. There's now 250 or 260 diseases for which we now have at least statistically associated associations. In most diseases, multiple places in the genome to look. And while it hasn't yet revealed the exact variants that are conferring that risk, it has greatly moved the ball down the field so that one can imagine now over the next decade or so teasing out a lot of these variants for a lot of diseases and knowing a lot more. But it's going to be complicated and it's going to be very challenging. But nonetheless, it's incredibly compelling to be able to accomplish this. One of the things that's very clear for dissecting out complex genetic diseases is that we're going to need to be in a position to sequence a lot of people's genomes to get complete inventories of each and every person, all of the variants in their genome, to figure out which are the ones that might be implicated for complex diseases. So that leads to the fourth step of 5 I want to review for you, and this is the ability to sequence whole genomes. Now, out of everything I've said, I would have to tell you that this has been the greatest triumph in genomics probably since the end of the Human Genome Project. Certainly technologically, I would actually say this is the greatest triumph in genomics since the beginning of genomics. And that has been the development of new technologies for sequencing DNA. This has, to date, mostly influences these first 3 domains, but trust me, with time, genome sequencing is going to really be influencing these clinical domains as well. The genesis of this really began the day the Genome Project ended, and that strategic plan that I told you about, where we made a declaration that we should go forward as a field and figure out how to sequence a human genome for $1,000, which was really quite audacious for us to say, because of the day the Genome Project ended, we had sequenced the first human genome, and it had cost $1 billion. And now we said, oh, let's come up with new methods that would eventually let us sequence $1,000. A complete genome. And in fact, the phrase $1,000 genome became a battle cry for the field, and it became a battle cry for venture capitalists and the private sector to get involved, and all of it's happened. We've given out grants, they've given money, and between academic groups and businesses that have been built up around sequencing technology, it has been remarkable what has happened. And I think we're once upon a time, it's one of the examples I alluded to earlier, once upon a time we said, oh, someday we'll be at $1,000 genome. That someday came far faster than we ever possibly anticipated. To give you a flavor of what I'm talking about, as I mentioned to you, that first human genome sequence, 6 to 8 years of thousands of people around the world working to deliver for $1 billion the first genome. Interestingly, the day the Genome Project ended, we asked people to go and calculate if you had to sequence the second human genome, how much would it cost? Even then, it would have taken those thousands of people around the world and all their laboratories 3 to 4 months to accomplish it, and it would still cost $10 to $50 million. Ask me today, using some of these fancy new technologies that I'm sure many of you have heard about. How long does it take? What does it cost? Well, today, I can take any DNA from any one of you in 2 to 3 days for something on the order of $4,000 to $8,000. We can get a complete genome sequence of you. In fact, by the end of the calendar year, this will be down to a day. We already know one of the technologies will be able to do it in a day. And this number will quickly get closer, $3,000, $2,000. It's going to quickly get to $1,000. If you ask me, am I worried about getting down to the $1,000 genome? Absolutely not. I have a lot of things to worry about. We will coast to a $1,000 genome in a matter of a few years, if even that long. That doesn't mean I don't have things to worry about. Because what I worry about, in particular, is this 5th step. Because the 5th step is really a reflection of how we have become victims of our own success. We were so effective at developing these technologies for sequencing DNA that now the genomics community, and actually I think the whole biomedical research community, finds themselves in a position where data generation is not what's rate limiting. It's data analysis that's rate limiting. Because, and this is affecting all the areas of these domains, and which is why we're so concerned about it and really trying to hit this head on if possible. We've created this bottleneck in genomics because these technologies seem to spew out data quicker than we can assimilate it, sort of like trying to drink water from a fire hydrant or from a fire hose. It's just overwhelming us, and that's very new for us. Genomics, people may have thought, was a big data enterprise, but it really wasn't until very recently when we've been able to generate data so prodigiously. And it's something that, all the major journals are recognizing, talking about big data as being sort of the bottleneck, where once upon a time that was only true for particle physicists and for astronomers. Now it's true for biologists. And it's a difficult challenge, but one that certainly the NIH, not just my institute, all of NIH is facing this, and in fact, we are really aggressively trying to pursue this. Recommendations just came in from a task force, essentially, that the NIH director set up, a working group that advises him, and there's a number of things that we're going to be pursuing. The bottleneck has various flavors associated with it. There's sort of the classic hardware issues of having enough processors to store data, processors to analyze data, hard drives to store data, enough bandwidth to push data through the internet. But there's also problems. We don't have software tools that allow all investigators, not just the most genomic savvy investigators, all investigators to analyze large data sets of genomic information. And one thing that certainly we're talking a lot about is how are we going to train the next generation of scientists? How should we be training the next generation of physician scientists? How should we train the next generation of translational scientists? They need to be facile at big data analysis. And we need to think about how to prepare them for that, and that might require some changes to some of our training programs, certainly something we're talking about. But the bottleneck in genomics and data analysis is not just at the computational sort of turning the crank side of things, it's also in understanding what it means. We are capable to use these new fancy technologies for reading out DNA, and we could take a genome sequence of any individual or any of our clinical subjects or any of us. We can slog through it, and we can even generate that list of variants, those 3 to 5 million variants that exist in each and every one of us or our clinical subjects. But right now, today, for the great majority of those variants, we stare at them and we have absolutely no idea if they're even meaningful or not, or whether they're just neutral or silent or whatever word you want to use of not being biologically important. But in fact, we're sequencing genomes of lots of clinical subjects now as part of research endeavors. The truth of the matter is when we round down those patients in the morning and we look at that list of variants, we feel like we don't really know what most of those variants mean. It's the reason why Harold Varmus, who previously was the director of NIH, now head of the Cancer Institute, wrote a commemorative article about the human genome, but admitted even in 2010 that physicians are still a long way from submitting their patients' full genomes for sequencing, not because the price is high, but because the data are difficult to interpret. That's the situation we find ourselves in now. A good friend and colleague of mine, Elaine Martis, humorously wrote recently how we have a $1,000 genome, but we're going to have a $100,000 analysis. And sort of sad but true. We can't be in this position. We have to solve this, and we will. So those are the 5 steps I wanted to tell you about briefly. There are others that are very much relevant, especially out in these more clinically domains that we're going to be inching our way towards. And you're thinking about them, and you know which ones, some of them, new diagnostics. We want to use genomic information for better diagnostics. We want to use genomic information and what we learn about the biology of disease for developing new therapeutics and new preventative strategies. But I also want to remind you, I am convinced we have steps we haven't figured out we're going to have to tackle. So I thought I would do, having sort of given you those 5 steps, let me just spend about 5 minutes or so, because I promised you I would, telling you a little bit about the future, as much as I can predict the future. But I can certainly tell you some of the things that NHGRI is doing, as we see as a priority, and we know others in some cases are joining us for some of this. In predicting the future, let me continue with the strategic plans framework and tell you about what I think sort of the next decade is going to bring and then the decades beyond that by filling in this density plot. If you ask me, what is the next decade going to look like with respect to where is the action going to be? What's the center of gravity of these accomplishments? I think it's going to look something like this. Notice I'm not saying we are going to fully implement genomic medicine in the next 10 years. Absolutely not. It's going to take decades to get us there. Medical care, health care, very complicated. That said, I absolutely do believe that the next decade is going to bring remarkable progress in understanding how genomes work. I think we are going to continue to really understand mechanistically some complexities of the human genome. And I also think hand in hand with that, as I sort of alluded to with some of these advances in rare genetic disease research, common genetic disease research, you're going to see significant advances in our understanding about the biology of disease. With that will come more home runs and more activity out here in the clinical domains. Realistically, I think the center of gravity where we really see the accomplishments in these clinical domains are more likely to take place beyond 2020. I think we will inch our way there, and I believe it's absolutely coming. But I do believe it's not going to come in the next few years. Well, how are we going to even accomplish this? How are we going to, in particular, see major advances in understanding the biology of disease? Well, I will tell you it's going to happen because we are going to sequence a lot of people, a lot of their genomes, especially individuals with and without defined diseases. It's going to involve not just the people shown on this slide. Those are only thousands. This is tens of thousands. That's not. It's going to be hundreds of thousands of individuals. We already know at NIH, for example, we already have commitments in funding from the various institutes for projects that will deliver probably upwards of 70 or 80,000 human genome sequences over the next few years. And that's just the United States. There's no question around the world. There's many, many others. And there's also no question that if you look 5 years out, I'm sure we're dealing with hundreds of thousands. Now, the work we're doing is all clinical research, not clinical care yet. It's clinical research at major academic and government research facilities, research hospitals, such as the one at NIH pictured there. Increasingly, this will be done as part of thinking about how clinical care might capitalize on this information. But again, I think for a long run, for the long term, it's going to be within a research framework until we better understand operationally how this is actually going to happen. So you will see the databases will fill and fill and fill with lots of human genome sequence data. I really want to point out the other challenge we're tackling at NIH is we want to see this data aggregated across all these studies into common places that all people can compute across, not to have the individual investigators who generate it just have it for themselves. We want the genomic data, we want the phenotypic data, we want healthcare information. As much as possible, there's a big push at NIH now of data sharing, as there always was in genomics, but increasingly for these disease studies, if we don't get the numbers way up, we may not get the statistical power to tease out all these genetic contributions for disease. So one of the things we are emphasizing now increasingly is figuring out how to get data from all the different studies under one roof, if you will, for cross-analyzing and have the worldwide community of scientists have access to that data to think of their creative ways of doing the data analysis. In terms of specific areas that we're investing in at NHGRI, well, it all is around these new sequencing technologies and sequencing human genomes. One of the priority areas we've just launched has to do with Mendelian diseases. We, as I told you earlier, we now know the genetic basis for 3,500 of them, but what I didn't mention here is that's only about half of the pie. But there's still the remaining disorders, thousands of them, either known to be Mendelian or suspected to be, for which we don't yet know the gene. We think now is the time to really press the accelerator and take advantage of these sequencing technologies that would allow us to accelerate the pace of discovering these. So we have set up several centers who are going to be an intake for worldwide efforts to try to get these things sequenced very quickly and figure out if we can use the power of modern genomic technologies to elucidate the genetic basis of these remaining rare diseases. Similarly, we're going to be doing many projects on complex genetic diseases, shown here as a simple graphic, where it simply is pointing to regions of the human genome that have been statistically associated with important human complex diseases. And now is the time, we believe, for some of them like Alzheimer's and various forms of mental illness, cardiovascular disease, diabetes, and so forth, where there will be a very significant use of genome sequencing to try to get complete inventories of variants in these individuals and try to figure out at a much more refined scale which variants are actually the ones conferring risk for disease. Among some of the earliest, I think, triumphs you're going to see clinically are going to be advances in cancer genomics. Cancer is a disease of the genome, and many projects around the world, including here in Australia, are pursuing this by using genome sequencing approaches to inventory the variants that exist in large numbers of cancer specimens. At NIH, our program is called the Cancer Genome Atlas, but like I said, under the umbrella of the International Cancer Genome Sequencing Consortium, there are many countries involved studying now dozens and dozens of different kinds of cancer because of the recognition that this might prove to be some of the earliest triumphs in genomic medicine in the cancer arena. And I would actually agree that I think that is likely the case. There are other areas we are thinking about and that we're already investing in the near-term future. You will see things. Certainly the area of pharmacogenomics, knowing now, increasingly learning that the genetic basis of drug response comes down to some of these variants, and we can tease them out, figure them out, and then in some cases, as is already the case for several medications, standard of care involves genetic testing prior to making decisions about which medication to give a patient. A more complicated arena, but one that we're just going to start a granting program in, is to do research to start to see the issues and understand the issues associated with the future of newborn screening. Newborn screening done in Australia and in America is one of the first encounters an individual has to genetics. It's complicated, it's ethically charged, but at the same time, seems like what we do now is wonderful, but is probably a small tip of the iceberg compared to the kinds of things we might be able to do in the future. And what that future needs to be studied first to figure out what that might look like. And if we were going to do whole genome sequencing, for example, as a possibility very early in life, and what's the right ethical framework and practical framework and technical framework to try to pursue that. And so we're starting a granting program to explore this in the next few years. Similarly, we recognize that this is overwhelming in terms of how information is being generated, even among the scientific community. And yet as this gets deployed from scientists to patients, or scientists to healthcare professionals, and then of course from healthcare professionals to patients, those are major leaps that have to be pursued. And we need to do this in a very thoughtful way, and the thoughtful way has to be built on a foundation of research. And so we are also launching some research programs that will help to start to frame out what a clinical genomic information system might look like, which would help practicing physicians in particular. And of course, a lot of this might integrate with electronic health records, which seem like a natural home for genomic information. But increasingly, we need to think about convenient tools that we can make available to practicing healthcare professionals to quickly look up what variants they might have in their patients and what they do about it. They don't have time to go read the literature. We need to have it be very well curated and sort of standard of care practices made available for quick lookup. And we have no idea what that's going to look like. We need a research foundation on which we can build it. And of course, beyond that, there are many other issues I'll put under the general label of genomics and society. And here, patients come to the forefront in thinking about how are they going to be dealing with all this information? What do they want? What do they not want? How should we be telling them things? And so, as our institute has been involved for many years in studies of the ethical, legal, and social implications of genomics research. There are many issues we are pursuing, being mindful of many societal dilemmas that we're going to have to face, and we want to have, again, a research foundation by which we can make decisions. So that's pretty much what I wanted to tell you. I do want to remind you of one last thing before I end, and that is that this progression is very logical, and this progression is very straightforward when you represent it in a PowerPoint slide. But I don't mean to imply for a minute that making this progression is easy. It's not. It's going to be very, very challenging. There's a lot of reasons for that. One of the reasons for that is the recognition that as a community of genomicists, myself included, most of us sort of grew up and have expertise in sort of basic science. But we've been so successful, especially technologically, and the opportunities have been so compelling, we're quickly finding ourselves now in the translational science arena with human subjects and disease. We're thinking about medical science and so forth. And I will also tell you that we're learning that to really traverse all 5 of these domains, we even have to start thinking about implementation science, of thinking about really getting data that's going to help figure out whether or not things are practically going to improve the effectiveness of healthcare or not. And even translational science is a stretch for many members of our community and many members of my institute. When you get out here, we're really students of this. So we recognize this is going to be hard. It's compelling, but nonetheless, this is a big challenge because, in fact, in many ways, it's traversing a lot of landscapes that we are not necessarily so familiar with. So with that in mind, let me just end with a quote from Winston Churchill, because I think it's sort of fitting for this, where he once said that a pessimist, which genomicists are absolutely not pessimists, but a pessimist sees the difficulty in every opportunity. I think genomicists are optimists. We see this progression, and we see this as being hugely difficult, but we see it as an incredibly important opportunity, which is why we're pursuing it so aggressively. So I will stop there, and again, I'm happy to have any discussion and take any questions from you. Thank you.